good afternoon, everyone, to this afternoon session of the Wednesday afternoon lecture series here at the NIH. For those of you who don't know me, my name is Anirban Banerjee. I'm in the intramural program of NICHD. And this talk is being hosted by the Chemistry Scientific Interest Group. When I was a early graduate student being christened into the chemical biology ward from a pretty straitjacketed chemistry curriculum, I remember in our chemical biology lecture at Harvard, our professor, young professor David Liu, told us about this discovery where someone had designed a new substrate instead of ATP, a synthetic analog, and generated a mutant kinase with the idea that this is essentially an orthogonal ligand substrate pair, if you will. And there are a few things that the first time you hear them, they kind of explode in your mind and they stick with you all your life. And this whole idea of orthogonal ligand protein or enzyme discovery is one such thing. And it's a real pleasure and honor for me to introduce our speaker from whose lab this work originated. And that was 20-something years ago. Kevan was already a superstar in chemical biology. And he has sort of gone along that track. So we have to coin a different term for folks like Kevan. It's more than superstars in chemical biology, but you understand his impact in the field. And I think it's important to say two other things. One is that Kevan, someone who's been an extremely kind and widely known mentor, who's mentored an amazing number of trainees, and the impact of a scientist beyond their science is, of course, the trainees that they send out into science. And Kevan's trainees have gone into chemical biology, neurobiology, developmental biology, you name it. Kevan's research now focuses on essentially new small molecule tools and drug candidates targeting protein and lipid kinases, and really encompassing all the tools of synthetic organic chemistry, protein engineering, structural biology, biochemistry, and cell biology. And it intersects with neurodegenerative diseases, oncology. And one aspect I think that's particularly special about Kevan is that he's really been very intimately involved in translating these discoveries all the way to making a new drug. Kevan received his BA in Bachelor's in Chemistry from Reed College in 1986. He received his PhD in Organic Chemistry at UC Berkeley with Pete Schultz, where I think he worked on catalytic antibodies. And I think that probably spurred his postdoctoral choice, where he was a postdoc at Stanford with Professor Chris Goodnow. And he started his independent career in the Department of Chemistry at Princeton before moving to UCSF, where he's now. He moved in 1999. He's now an investigator of the Howard Hughes Medical Institute and a professor in the Department of Cellular and Molecular Pharmacology at UCSF, and also a professor at the Department of Chemistry at UC Berkeley. And not surprisingly, Kevan has received an amazing number of awards. In fact, I think the bio that he handed to us stopped at 2002, probably because he knew that we wouldn't be able to manage with all the awards that we have received. But I will still mention a few. So he was a Pew Scholar. He was a Searle Scholar. He was an Alfred P. Sloan Foundation Fellow. He received the Eli Lilly Award and the Protein Society's Young Investigator Award. He is a member of the National Academy of Sciences, the National Academy of Medicine, and the American Academy of Arts and Sciences. Thank you for coming to the NIH, Kevan.
Oh, thank you, Anirban. Thank you for that very nice introduction. It's always fun to hear about some of the early work, and great, thank you very much. And it's really great to be here, and it's a real honor to give this lecture. It's very special also to do it here at the NIH. As you'll see, a lot of the early RAS work was done right here. And so I'll take you through a little bit of that because it's so foundational. And I think without that, none of the things I could tell you about would have been possible. So first off, I do have the disclosures from several companies that have licensed compounds from the lab. The most relevant is from Araxy's Wellspring, which is at the last one on the list. So I think all aware that cancer is a genetic disease, and we get mutations in our chromosomes, and that give rise to selection for growth. And I want to break it down into sort of cancer into one slide, if that's possible, and give you a high-level picture of where we're going in our lab, and I think the field is also going. And so we know that proto-oncogenes become mutated, and the most frequent oncogenic mutation that occurs over and over is in the K-Ras GTPase, which I'll be focusing on today. And this gives rise to constitutive activation, constitutive binding to GTP, turning on many downstream effectors. And I just show the RAF MEK pathway here as one of the key determinants. And that gives rise to a signal that is growth factor independent and anti-apoptotic, pro-proliferative, and things like that. Now, if we just have cells that have one of those mutations, we don't have a cancer, and that's because we have fantastic protection mechanisms exhibited by the tumor suppressors like p53 that sense oncogenic stress. When p53 recognizes that stress, becomes a very active transcription factor, turns on hundreds of coordinate genes that then arrest the cell cycle and send the cells into apoptosis in some cases, and the cell is in a senescent state. So at that point, the oncogenic mutation is not driving proliferation. But this cell in a senescent state is also subject to a mutation and selection, and often p53 is mutated, losing its function, losing its tumor protective effect, and then the cell starts to divide. And to give you an idea of the numbers of patients, RAS is the most frequently mutated oncogene. p53 is the most frequently mutated tumor suppressor. And if there are about 18 million cancer patients worldwide every year, about 9 million will have p53 mutations, and about 2.5 million will have K-Ras mutations. So it's a tremendously frequent lesion that occurs to drive many, many cancers. Now, once the cell is rapidly proliferating, the oncogene is mutated, tumor suppressors are lost, then there's lots of errors during the cell cycle and the rapid cell division. And so we pick up even more mutations besides the oncogenes and the tumor suppressors. And this symbol just sort of is meant to emphasize that we get patients, tumors, get private mutations, mutations in genes that don't necessarily help selection, but they just accumulate over time. And the next chance for the body to fight the cancer is the immune system. And so these mutations that occur, either that are promoting growth or just bystander mutations, get processed into peptides, and then these peptides become presented through antigen presentation on class 1. And because these are mutations that occur only in the tumor cells, then they're potentially recognized as non-self, and patients' T cells can see those if the T cell receptors have the requisite specificity, and then the T cells can kill the tumor cells. Now, again, this case, the tumor can fight the immune system, and we know that from upregulation of things like PD-L1 that then signals to checkpoint receptors on T cells like PD-1, and we know that that then short circuits the T cell from killing the tumor cell. But of course, because of the remarkable advances from the checkpoint therapies and the discoveries from Jim Allison and others, we know that inhibiting PD-1 or PD-L1 can take the brakes off T cells and let them kill the tumor cell. And these are fantastically powerful therapies. The problem is that the mutations that sort of drive the oncogenes and the tumor suppressors over here are often poor peptides to be presented or are poorly recognized by T cells to allow killing of the tumor cell. Many of the antigens that people have identified that are suitable for letting the T cell kill are really from these bystander mutations or the private mutations. So it's difficult to get the right recognition. Now, here at NIH, Steve Rosenberg's group and the groups in that division have made fantastic inroads into identifying patients' T cells that do see mutants that are in hotspots like the K-Ras peptide, and they've shown some fantastic responses of patients when you infuse back the right T cells. So these are really fantastic signal-finding studies. But what we really have been working on is to try to leverage basically therapies that inhibit the oncogenes, reactivate tumor suppressors, and somehow avoid the immune evasion that occurs in tumors. And I think if we can find very specific ways to do that, we can combine these therapies, and then we might get farther towards deeper responses. So that's what I'll tell you about today. I will talk mostly about inhibiting K-Ras. I won't talk about reactivating tumor suppressors like P53 that we've done, but I will talk about how to leverage some of the antigen presentation pathway to sort of get deeper responses. Okay. So the title pointed out that K-Ras was considered undruggable, and I want to give you a little bit of a history of K-Ras from its discovery and what made it a challenge for identifying the first drug. So K-Ras here now you see is a GTPase. On its C-terminal tail, it's lipidated with a farnesyl group that I'll go into. Once it's at the membrane and it's constitutively active, and it turns on the signaling pathway that I showed here. So it was first discovered as a human oncogene in 1983, and until 2021, there were no direct inhibitors approved for patients. And if you want to sort of compare that to what a successful druggable protein looks like in terms of timeline, the BRAF mutations were discovered many, many years later in 2002, but as soon as they were discovered, 9 years later, there was an approved BRAF inhibitor. So from discovery of the oncogene to approval, it was only 9 years versus decades in the K-Ras case. And the first challenge that was apparent was that the ways we make kinase inhibitors is by orthosteric inhibitors that compete ATP. And so you would think that because K-Ras is a GTPase, binds nucleotide, make a GTP competitive nucleotide. However, the affinity of K-Ras for GTP is 20 picomolar compared to the Km for ATP for kinases, which is 20 micromolar. That million-fold difference means that there will probably never be a GTP competitive K-Ras inhibitor. And this discovery, it was Herculean even to measure this number. This was done by Fred Wittinghofer and Roger Goody in Dortmund, and they had to almost denature the protein and measure kinetic binding because you couldn't do equilibrium sort of numbers to measure that. So the first challenge is the most obvious druggable pocket is off limits. Okay. So once that was realized, then other strategies began to be attempted. And the real first one to make it to the clinic were attempts to block the farnesylation of K-Ras because, as I'll show, that is required for K-Ras oncogenesis. So going back to the discovery of the RAS genes, they were done by retroviral isolate identification, passaging in animals, and then harvesting the viruses. And so HRAS was the first RAS gene isolated from Jennifer Harvey, and then Werner Kirsten identified K-Ras several years later. And some of the very early foundational work was done by Mariano Barbacid when he was here at the NIH, and by Doug Lowy in identifying the human gene homologous to the viral gene that was isolated. And I think a really key paper from Doug's lab was this one that showed that the serine, that the cysteine that becomes farnesylated, if it's mutated to a serine, so cannot be farnesylated, blocks all oncogenesis, done by classic soft agar experiments. So this one experiment really lit up everybody's excitement, because although it was difficult to think of a drug that could bind to the protein itself, we knew that the farnesylation was absolutely required. So shortly after that, a lot of work was done to identify what the lipid was. It was turned out to be a farnesyl group. And then farnesyl transferase was purified. So then the target that you would inhibit to prevent farnesylation of RAS was identified. And then quickly after that, inhibitors of farnesyl transferase were developed. So once that cysteine to serine mutant was made and the phenotype was clarified, then it was a mad rush to identify inhibitors of RAS farnesylation. So that was a really amazing decade, I would say, from the understanding that it was what the oncogene was and to a preclinical candidate. So what I want to tell you about is the experience with the farnesyl transferase inhibitors. So here's what the symbols will look like. So here's K-Ras was really the first oncogene, human oncogene discovered. So a lot of the studies were from the bladder cancer cells where K-Ras was transforming. And there's the G domain that binds the nucleotide. And then there's a flexible tail that has the cysteine that becomes farnesylated by farnesyl transferase. And so this isoprene unit displaces the pyrophosphate, and it becomes labeled. There are a few other steps of processing. You'll notice this is a tetrapeptide, but then in the final processed membrane localized form, it terminates in the cysteine, but it's carboxymethylated. That won't be so important here, but it's a very regulated processing step. And so, as you can sort of tell from my drawing of farnesyl transferase, it's got a very nice potential drug binding pocket. It's where the farnesyl binds. And inhibitors were possible to be made very quickly. And farnesyl transferase inhibitors absolutely prevent K-Ras localization to the membrane and kill cells. All the preclinical models said that. It worked beautifully. Now, take a step back and look at the human cancer picture. And there are mutants in K-Ras and mutants in K-Ras. And they have very similar G binding domains, but they have different tails. So K-Ras has a different tail than K-Ras. K-Ras comes in two splice variants, K-Ras 4A and 4B. And this sequence has multiple signals on it for multiple post-translational modifications. So if I add those sequences, you see that K-Ras and K-Ras 4A look quite similar to each other, but the K-Ras 4B has a polylysine added on its C-terminus. And you'll see that K-Ras and K-Ras 4A have another cysteine upstream, and that actually becomes palmitoylated in K-Ras and K-Ras 4A. So what I told you is farnesyl transferase inhibitors block K-Ras localization perfectly. But now there's an issue of which of these isoforms is the most important for human cancer. And what you can tell is that the frequency of K-Ras mutants are just 1%, but for K-Ras, it's 25%. So now we're interested in whether these can be prevented from membrane localization. And then we have to wonder is which alternative splice form is more important for cancer? And this is where the big problem came. The K-Ras 4B with this alternative polylysine sequence is the most important for oncogenic signaling. And because of that, and because of this different sequence, the entire farnesyltransferase approach for K-Ras mutants failed. And why was that? So the drug works well, inhibits farnesyltransferase. K-Ras does not get farnesylated, absolutely true. But then biology had a backup for K-Ras4B, and that is geranyl transferase. So one additional isoprene, different enzyme, but this enzyme can recognize the 4B tail. It doesn't recognize HRAS or the 4A tail, but it recognizes the 4B tail. And so all of the K-Ras mutant cells then allow localization, even though they had a fantastic drug. And so late in the 90s, this report and others really pointed out that the difference in the tail completely allowed K-Ras4B mutant cells to bypass farnesyltransferase inhibitors. And multiple companies had drugs in the clinic, thousands of patients enrolled, very few responses. This is at least right now, what we believe is the reason for all of that failure. So between the difficulty of the nucleotide pocket and this incredible rush to farnesyltransferase inhibitors, which ultimately failed, that really led to the problems and the view that K-Ras was going to be undruggable. So let me then go back up and tell you about the glycine 12 mutation and why it occurs so frequently and what does it do to the RAS cycle. So RAS is a nucleotide that switches between two conformations, and you'll hear me talk about this quite a lot. In the GDP state, it's off. In the GTP state, it's on. And by on, I mean that these two flexible loops on RAS, these two switch loops, form a conformation that on the surface that is compatible with downstream PI3 kinase or BRAF binding. And so when RAS is in the GTP state and it's at the membrane, it localizes these cytosolic factors up to the membrane, and then they turn on the cascade. Now, in order to make the cycle go from, in the wild type case, from off to on, and I told you the affinity is picomolar, well, there needs to be some way to catalyze the release, and that's done by this guanine exchange factor that comes up to the protein and essentially strips out the nucleotide. And then once free nucleotide state is present, then GTP can bind and turns on the cycle. Then there's a timer, and there's a fast off and there's a slow off. The fast off is through a GTPase activating protein that comes up to the complex and helps accelerate GTP to GDP hydrolysis. The slow off is intrinsic hydrolysis that happens on the free protein. And the key structure that really revealed all this was from Fred Widdinghofer's lab that showed that when GAP comes up to RAS, it pushes an arginine to fill the catalytic site and accelerate hydrolysis. And guess what's right at that interface is glycine 12. And so that small amino acid side chain is sitting right at the catalytic residue donated by the GAP protein. And so any residue other than glycine pushes the arginine away, ruins catalysis, the protein stays in the GTP state, turns on the downstream signal. Okay? So this is really the most frequent single amino acid change in any oncogene, and it occurs over and over again. But because it's a steric problem, really any amino acid at that glycine position can cause oncogenesis. And what's fascinating is that across the different diseases, these are the 12 most lethal cancers typically in a given year in the United States, starting with lung cancer. Incidence is in green, mortality is in yellow. In blue are the fraction of patients that have a RAS mutation upon presentation. So the key common K-RAS-driven diseases are lung, colon, and pancreatic cancer. Pancreatic cancer in particular, because almost 95% of patients will have a K-RAS mutation. It's the prototypical K-RAS-driven disease. The glycine 12 can be mutated to any number of single base pair amino acids that will switch over the codon, and in pancreas, it's aspartate, then valine, then cysteine. In colon, the spectrum changes a little bit, and you get a growing in of cysteine and a decrease of aspartate. And in the smoking-induced cancer, the dominant mutation is cysteine. And when you look at all of the frequencies and put them all together, the order is aspartate, valine, cysteine, and then several others, serine, arginine, and alanine. And so this kind of is still a little bit of a mystery. We don't quite know why you get the spectrum. It has to do with maybe slight differences in biochemical rates of hydrolysis when these different mutations are present. But whatever the case, as a person trying to look at the druggability of the complex, we realized, well, we have a number of amino acids we could target. And whenever we see a cysteine in a protein, we immediately chemically think of a covalent bond because it's the most nucleophilic residue and it's one of the rarest amino acids. So it has a very opportune sort of chance to make a covalent bond to it. So we set out to make a covalent bond that would bind to the cysteine. Now, one feature that we sort of get for free if we make a covalent bond to a somatic cysteine mutation is that the drug that we identify will only inhibit the signaling in the tumor cell and not the rest of the body. That is a whole different thing than most protein kinase, almost all protein kinase inhibitors we have. They inhibit the proto-oncogene and the oncogene. So by leveraging the chemical uniqueness of cysteine and the fact that the cysteine is only in the tumor, we really had a chance to make not only the first K-Ras drug, but one that would be highly selective for the tumor. So it came very, very attractive to take this approach. So we solved the APO structure, and we see the cysteine right here at the 12 position with GDP bound. Here are the two switch regions. One of the switches, switch 2, has a disordered element that was not visible in the GDP structure. And then we needed an approach to identify a starting drug. And we took an approach that was pioneered by my colleague, Jim Wells. This is the advantage of being at a certain place and knowing what your colleagues work on and sort of hearing about their discoveries and trying to see when they could all work together. So with Jim, he had developed this really powerful approach for discovering covalent ligands for cysteine-containing proteins using disulfide chemistry. Disulfide chemistry has the advantage that it does rapid exchange reactions, and so it can do both forward and reverse reactions, so it's a thermodynamic kind of search. And then the ligands have small drug fragments on them, and if one of them is complementary to a pocket near the cysteine, then the disulfide bond that's made will be quite stable. It'll be somewhat resistant to a non-specific thiol like beta-mercaptoethanol. So this strategy can be readily tuned by increasing or decreasing the BME concentration, and any molecules that are binding are detected by whole protein mass spec, so you know exactly that the molecule you expected is the one that's bound. So it has lots of features that are common sort of challenges for fragment-based drug discovery. So we used that approach that was pioneered in Jim's lab, and we identified these two hit molecules. Out of 500 molecules that bound to the protein, they bind to the K-Ras G12C protein. They don't bind to the wild type, and there's 5 other cysteines, so we knew it was selective. And then we began to do additional studies like the nucleotide specificity. This protein that we used was just harvested from bacteria, so it had already cycled to the GDP state. So we put the protein in the GTP state, and unfortunately, nothing bound to that. Now, remember I told you that the oncogenic mutation locks the protein in the GTP state. So the protein that's in the cell is the GTP state, and these two drugs don't touch that. You can see the labeling is almost non-existent. So we immediately put these aside. We went back to the collection with this form of the protein and asked, what do we get? 0. Not a single molecule touched that cysteine when it was in the nucleotide, the GTP state. So we basically decided that we had no other leads, so let's go with these and try to understand where it's binding, and even though this probably would never work even in a cell, let alone a patient. And so when we solved the structure, we realized immediately, first of all, that it was bound to this is the glycine 12 position, it was the disulfide, so you see both sulfurs, and then you see the drug binding in a beautiful pocket of the protein that we had never seen before in any of the existing APO structures, because it's underneath switch 2, which is half closed on the drug. And that really exemplifies that you sort of needed the drug to find the pocket. You couldn't look in the protein and find a pocket. You had to have both together. And it also explained why the drug doesn't bind to the GTP state, because it's binding in the pocket that the switch 2 would be in if there were GTP bound. So all of the things all of a sudden with that one structure really became very, very clear. So we then realized, in the slides that follow, I'll put a red dot on the atom of the drug that is attacked by the glycine 12 substituted residue. So in this case, the cysteine attacks this position, and you'll see that later. So we had a very early lead molecule. We knew we had the first pocket on the protein. Other people had found a pocket called switch 1 that was behind the switch regions, but that was even more shallow than this one. That was from the FESIC lab and the Genentech lab using SAR by NMR to identify leads. So we decided to continue to pursue this pocket. We know that the drug had very many fully rotatable bonds. And I showed you that pocket of RAS is very flexible. That's part of the pocket was not even ordered in the GDP state. So we needed to sort of optimize the drug, and that's when we started this company, Araxes, that elaborated many, many molecules and optimized for binding until we got to this very key molecule, which is much more pre-organized than any of the others, but still orients the acrylamide warhead and the affinity element that's back here. And once that was released, a lot of companies began to develop additional K-Ras G12C inhibitors, and they all share the same acrylamide and many of the same binding contacts, although each of them have slightly different flavors. This one is from Amgen, this one is from Mirati, and there's currently 10 other K-Ras G12C drugs in the clinic that are different flavors of these kinds of molecules. And before I show you the clinical data, this is just illustrating what so many chemists across industry can really do to add to the high affinity elements that we need for better and better drugs. So we've benefited a lot over the last 5 years from seeing many of the advances that people have done across industry. But the good thing was that even the first drug showed clinical activity. So this is the Amgen sotorasib drug in the approval trial, and this is a waterfall plot of patients that have lung cancer, that have G12C mutations and receive the appropriate dose of the drug. And this waterfall plot shows the change in tumor growth. On the left is the poorest response, on the right are the best responses, and you can see in green there are some complete responses. But the vast majority, about 80% of the patients, have what's called disease control. We don't have cures, but we have treatment benefit. And so this really, I think, opened the door to say that a direct K-Ras inhibitor could work. And although I'm skipping over a few slides that explained why, even though this molecule bound only to the GDP state, it still worked in cells and in patients. And that is because although the picture of the protein being locked in the GTP state was what we thought, it does cycle. And so what this drug waits for is the protein to cycle to the GDP state, then it latches on, and then it pulls it out of the cycle. So it's not ideal, not necessarily what we would have wanted, but the fact that that even works is exciting. So some of the drugs that are in the clinic now, the so-called second generation and next generation G12C drugs, are able to bind to the GTP state. They may be better than these. But this is a really, I think, a great start, and we have lots of room to improve. So with the G12C tumors in somewhat of control with compounds that are showing efficacy in the clinic and 2 drugs approved, we began to think of the other mutations. And we liked this covalent idea because, again, like I said, it gives you very, very good specificity for the tumor and it avoids the rest of the body. Now, at the time, in about 2014 and 15, we tried to jump from cysteine to aspartate. And you'll know that that's a much less nucleophilic amino acid. It's much more common amino acid, very big challenge. And we took everything we knew from about 6 years of work and applied it to covalent aspartate reactions. And the best we could do was a 5% conversion to the adduct. And that was where we sort of maxed out. So we began to take a step back and said, let's learn from these other residues that have nucleophilic potential, and then maybe we can carry that forward. So the one I'll tell you about that we've made farthest and learned a lot about is the serine. I also mentioned the arginine. We're still working heavily on the aspartate. So the warhead that worked the best for serine is this strained beta-lactone. And because of that strain, of course, we have the chance to release a lot of energy once the serine attacks and releases the alcohol. And that inspiration came from these natural products that inhibit and that bind covalently to serine and threonine residues, often in the active site of proteins. So the proteasome opens up this beta-lactone, this serine opens up this beta-lactone, and in palmitoyltransferase, serine opens up this beta-lactone. So these all seemed like they had the requisite chemistry. The problem was that these serines and threonines are the active site residue, which have vastly accelerated potential for nucleophilic attack because they're in the enzyme active site. So we didn't know whether we could translate that to a non-catalytic residue, the serine that came up in this oncogene. So we put the warhead on many, many different scaffolds, and the first one that showed really, really encouraging activity was this fused beta-lactone. And this element I won't talk too much about, but I'll show you some advances on that in the next few slides. This is the part that's binding reversibly in the switch II pocket and presenting this beta-lactone. And it labels 100% at 2 pretty high concentrations, but gives us good encouraging activity. But the most surprising thing about these weaker nucleophiles and these kinds of warheads is that they're incredibly sensitive to small changes. So if we just swapped the carbonyl and the oxygen, everything else is the same, we get almost no labeling. Or if we just add a single methyl group to the ring, we get very little reactivity. And again, methyl group on the dead one, we also get no activity. Now, we don't lose everything. If we shrink the 6-membered ring to a 5-membered ring, then we get activity back. So this SAR is just to sort of exemplify how tight everything has to be poised for the attack of something that is not as nucleophilic as cysteine. So that taught us that we could have a lot of frustrating molecules that don't do anything, but we still have to keep making the small changes, because if you were right here, you would think you didn't have anything, but as soon as you make the opposite one, you're ready to go. And so we solved the crystal structure of this bound in the protein, and this is only the second covalent structure to a RAS isoform, now the serine. And this one showed the serine making an ester bond, and then it opened up the alcohol. It makes really beautiful contacts with the rest of the pocket, including important elements like this conserved lysine that's not mutated in the cancer, but it's important probably for the chemistry to occur. So with that, we found the right warhead. Now the question is, I told you that we couldn't change the warhead very much, right? Every time we changed it, we lost activity. So how do you improve something if you can't touch the warhead? And that really comes from reversible affinity improvements. And so this was the rate of reaction of this first molecule. And you can see after it's got about a 50-minute time to get to half occupancy. And if we make changes that came from a company called Mirati that had made many, many optimizations of this for reversible binding to other alleles, as we make those changes, like this 5-membered ring to what's called the butterfly amine, you see improvement, big jump, and then making this unsaturated, we get an even bigger jump. So the great part is now we can start really dialing in affinity for these RAS ligands because so much chemistry is being done, we're getting lots of ideal contacts with this one pocket. And so that is what we were able to leverage. And now when we go into cells, we can see that those early molecules like 3 and 4 that don't label very fast, they don't block RAS GTP levels, but the rapid on molecule does very well. And you can see that in a panel of G12S cell lines, the phospho-ERK signal is always diminished. Cells that don't have G12S are not sensitive. So again, it's this mutant specific only for the serine. Really, really encouraging for our ability to dial in these mutants. And one thing I should have said is there's a whole other reason to go after G12S. And that is there are genetic defects called rasopathies that occur in patients. And in Costello syndrome, one of those rasopathies, the dominant mutation that comes is in HRAS, and it's from the G12 to S allele. So almost all those patients have that single residue conversion. So if we can convert this molecule, right now it doesn't inhibit HRAS in the G12S state because it has sort of a problem with one of the residues that's different between H and K-Ras. But if we could do that, then we would have a therapy that would inhibit just the disease-causing mutant that in those patients is in every one of their cells. So that's a direction that Xiang Zhang in his own lab at Berkeley, who did this in my lab, is taking. One question you might be thinking is, now that we've gone after a less reactive residue, when we put this molecule into cells, does it potentially react with all kinds of serines? And so we did a proteomics experiment where you can take a click version of the molecule and compete it off with a non-click version, and then look at which proteins labelings are competed. And this is in a G12S homozygous cell line. You see K-Ras comes up as a positive target that's competed by this drug. And then there's only 3 other enzymes. One of them is a phospholipase that has one of those active serines. So that was kind of an expected potential off-target. But overall, this is the kind of selectivity that we see on the G12C drugs. So we're able to get selective binding only to K-Ras G12S with only limited numbers of off-targets. Okay. Well, we also took what we learned on the serine and applied it to arginine. And G12R is a very interesting mutation. It occurs actually only in pancreatic, dominantly in pancreatic cancer. And because the arginine has these 2 amines, Xiang designed a double warhead that would react with each amine and make a 5-membered ring as the adduct. So we can even go after more unusual amino acids in a mutant-specific way as well. And then the big one we're after, of course, is the aspartate, and we're working very hard on that right now. And there's some encouragement, I would say, that's looking promising. So let's go back a moment in the last few minutes to the current clinical picture of the G12C patients. And there's kind of 2 kinds of issues going on. One is there are a number of patients that are really non-responders. They have the mutation, the drug hits the target, but something in the cell signaling is not sending those cells to apoptosis and not shrinking the tumors. The other problem is patients that are over here, they respond, but then they become resistant. Classic targeted therapy problem we knew all the way from the beginning of Gleevec. And so we'd like to treat both of those scenarios, and there's a lot of combination therapies going on right now to try to deal with that. But we decided we wanted to take a different approach and see if we could come up with a different strategy. And to tell you about that strategy, I want to tell you a little bit about the resistance spectrum that is coming out of the patients. And so this is a panel that has 4 patients listed. The top patient is a patient that all of them responded and then relapsed. The first patient responded and then picked up a mutation in the K-Ras G12C allele, this Y96C, that doesn't let the drug bind. So that's like a BCR-ABL mutation that prevents Gleevec from binding. That's kind of what we anticipated. But actually, the most common kind of change is shown here on these 3 patients, where the first column is the ctDNA before therapy and then after relapse. And what you see is a whole slew of mutations, and if you can read the labels, they're in the RAS pathway, including like BRAF mutations. So they're addicted to the K-Ras pathway, and you pick up new mutations in the RAS pathway to overcome the drug. But what's interesting is you still express the K-Ras G12C. It's not gotten rid of. And sometimes there are multi-copy amplifications of the G12C allele. So what that looks like is we have a patient with G12C. We give them a G12C inhibitor. It goes into the tumor, binds covalently, shuts off signaling, and then mutants occur downstream, and the pathway is back on. So even though the protein is there, the drug is still binding to it, it doesn't matter because the signaling is still on. So what we began to do, and I collaborated with my colleague Charlie Craig on this, is to try to leverage the covalent nature of the G12C drug and the mutant cell specificity to bring in the immune system to attack that tumor. So even though there's no signaling inhibition or diminished signaling inhibition, can we create a neoantigen out of the drug conjugate? And does the protein get processed and presented up to the cell surface? I know that's a complicated little approach, but what I'm going to tell you about is that, so we've got class 1 MHC, the G12C peptide is there, that's red is the cysteine, and the drug is attached. It came from the drug binding to G12C, as I've shown you, and now we're going to make an antibody that recognizes the drug peptide conjugate connected to an anti-CD3 to make a bispecific T-cell engager to kill the cell. So the first question is, does this peptide with the drug still bind to MHC? And we can do that by an MHC cell surface stabilization assay. And if we give the, if we don't give any peptide, this is a TAP deficient cell line, and MHC comes up to the surface and is not stable if it doesn't have peptides. So exogenous peptides can be added. Give a positive control peptide, MHC is stabilized, the K-Ras peptide stabilized, K-Ras drug conjugate stabilized. So we know it binds. Then Charlie's lab made the antibody from a human naive library that recognizes the drug, and then the subsequent antibodies we've made recognize the drug, the actual peptide sequence, and the MHC. And then we can ask, does this system allow killing of a cell that has a bypass resistance mechanism? So here's the G12C sensitive cell line with the drug and the bispecific T-cell engager. This is the killing that we see. Now if we put in a glycine 12 to valine, the drug can't touch that, so it inhibits the signaling produced by the G12C inhibitor, but now we still get equivalent killing. So what we've done is we've made a way to kill the cell independent of its inhibition of signaling. And that, we think, could be a great way to cross the sort of bypass mechanisms that tumors might develop to avoid killing by these drugs. And because the immune system is so powerful and T cells can have bystander effects, we could even deal with heterogeneity and a whole number of issues that we know are emerging in the clinic. And then the last slide just shows that what I've told you is that we've got a way to bring in a T cell to kill a tumor cell, but there's a lot of interest in radioligand therapy, and Mike Evans, one of my colleagues, is really an expert in this area, so we're putting on lutetium beta emitter to do that killing as well. And this won't require that a T cell is accessed in the tumor environment. It could be an immune desert, and this approach should still work. So we're really interested, as I pointed out at the very beginning, trying to leverage targeted therapies and immune therapies to try to sort of short circuit a lot of the bypass mechanisms that tumors seem to always have ready to avoid new drugs. So with that, I'll just tell you that two fantastic people in the lab that did the first G12C work were John Ostrom and Ulf Peters, a graduate student and a postdoc, and we leveraged the beautiful screening approach of Jim, Michelle, and Adam in the Small Molecule Discovery Center. Ziyang was the person who really persevered on the beta-lactones for the serine attack. And the G12R, he also developed that warhead with a current postdoc, Johannes Morstein, and a rotation student, Andrew Ecker. And then the covalent drug recognition was really a collaboration with Charlie Craig's lab. Postdoc, again, Ziyang worked on this with graduate student Peter Owader in Charlie's lab. And then the radioligand therapy is with Mike Evans and his postdoc, Apurva. So thank you very much for your attention.
What is the mechanism for GLN61 and ALA59 mutations? I saw them on NRAS in myeloid neoplasms.
Give me the two numbers again in NRAS.
Well, if the fonts are correct,
Yeah. Probably.
GLN61.
Yeah, glutamine 61. Okay.
Okay,
Yeah.
and ALA59.
Oh, wow.
Yeah.
Those are fascinating. ALA 59 A to T was, I think, actually discovered here at the NIH in one of the viral isolates. And that A to T mutation is oncogenic in the context of a glycine 12 to serine mutation. And exactly why that causes the mutation, oncogenesis, is not exactly clear. But it was a fantastic sort of point because the threonine becomes phosphorylated by the K-Ras GTP. So it was it was a little bit of a detour in the RAS history because people thought it might have been that RAS was a kinase because of that threonine. But glutamine 61 is like the most severe mutation you can find because it loses one of the catalytic bases for intrinsic hydrolysis and gap hydrolysis. So it's fully locked in the GTP state. And that's the common mutant in NRAS, which I didn't talk about too much.
Okay. What role does the rate of K-Ras turnover play in the success of these covalent inhibitors?
Great question. I believe its turnover is very important because we know they only bind to the GDP state. The approach that the explanation I gave you was that intrinsic hydrolysis is responsible for the turnover. But in work from Pirolito's lab at Sloan Kettering, he found an alternative gap protein from the one I showed that seems to support hydrolysis. So there might be alternative gaps. They're hard to find in the cell. So turnover is very important. I'd say we don't fully understand all the ways it can get turned over.
Okay. I'm going to go, and you go, and I'll have one more from online.
So thank you for that beautiful talk. And the last part with the T cell targeting blew my mind. On the but does that help with the classical view of evasion or of resistance, where the mutation is in K-Ras and blocking the binding, as you saw some?
Excellent point. Yeah, for that first patient in the panel that got the mutant that doesn't allow the drug to bind, that approach wouldn't work. Totally right. Thank you for pointing. Yeah, that would avoid. Yeah.
Thank you for that lecture. You made something extremely complicated understandable. I really, really enjoyed that very much. What I'm still not understanding is why is the K-Ras 12, G12, a target for mutation? Is it something about the chromosome structure, and why is it different in different tissues?
Gosh. Yeah. Okay, I'll get to that second. This is like the more fundamental biology question that has been perplexing from the 80s. And I just came from a RAS meeting up in Philadelphia, and it was the topic of the discussion. So it just tells you it's a great, great question. I think the subtle differences in the amino acids and how the cycling is affected creates a different flux in the pathway. And because first time the cell is exposed to the oncogene, it has to deal with that stress and turn on the p53 response to survive long enough to get the next hits. I think we think that a lot of that specificity is controlling that. So other background mutations are important. It's kind of why we don't see Q mutants in K-Ras. It's probably too strong of a signal, and then the cells die, and they can't be around. It's still a fascinating question.
Lots to be done.
Yes, yes.
Thank you so much.
One over here.
Oh, you asked the G12V. Yes, there are.
Not the same thing I saw. I'm
No,
sorry.
it's a little different. No, it was
Okay.
close. But the G12V can be targeted, and one of the drugs I didn't talk about, I should have a slide on all the new drugs that are coming out. They're coming out very quickly, is a reversible K-Ras binder called MRTX1133 that we talked about at lunch with some of the trainees. And that allele can be bound by that molecule. Now, what the selectivity is going to be and on all of that, but we do have a grip on that now, I would say. Mm-hmm.
Yeah, really fantastic talk. Thanks for sharing. I thought the formation of the serine ester was really fascinating. I was wondering if you had seen any evidence that that ester could be resolved by either a different residue within the same protein, like acyl transfer, or whether a different protein could take up that ester.
Yeah, that's a great question. Yeah, thanks, Ross. We thought that this lysine would attack that exactly, and then via transfer, we never got evidence for that. So it is fascinating. It'd be kind of cool, actually, now that you mention it, to maybe make a gap protein and try to get the gap to kind of stick onto it. Maybe that's why I hadn't thought about that. That's a good idea.
Thank you.
Yeah.
So that was fascinating. I had a question about sort of the general sort of strategy of covalent labeling of cysteines, because from a very naive perspective, if I hadn't seen this work, I would have thought it's impossible to get a cysteine reactive agent in a cell selectively reacting with a protein. Mm-hmm. But clearly you can tame that reactivity by other parts of the molecule.
Yes.
And multiple engagement is what then makes that reaction possible. So I guess what I'm saying is that, do you think that you or the field has exploited this almost sort of caged reactivity and controlling it with conformational flexibility to the fullest extent possible?
Yeah. No, that's a great question, Anirban. I think there's like multiple layers of that. The first layer is that, this is the warhead, but this piece is pretty big. And so there might be a lot of cysteines that would react with this, but because it's attached to something steric, it doesn't get there. So that's one thing. The other thing is it can be tuned, and the difference between this molecule and this molecule is kind of interesting. You see that this warhead has alpha fluorine, which detunes it. The reason they needed to do that is because this molecule, they had this cyano group that we actually still don't quite understand, reactivates this warhead. So if you put this cyano over here, this drug would be somewhat promiscuous. So in order to keep that cyano, they needed to detune. So we're starting to get really careful, unbelievable, selectivities going. And I forgot the third thing I was going to highlight. It's a lot of pre-organization. I mean, one of my favorite aspects of this pre-organization is this molecule. This bond can be resolved. They're atrope isomers. You could see that's restricted motion because double diortho substitution. This particular one where the phenol is out of the ring is the active one. Wow. If you made it with the phenol in the back of the ring, doesn't touch RAS. Unbelievable. So these things are like, yeah.
Hey, Kevan, great talk.
Hey, thank you.
I was curious about, linking kind of, the two parts of your talk. I mean, you talked about the, some of the reversible inhibitors that are now coming online. What do you think about the prospects of using those as degraders to prime up the immune system, right?
Yeah.
For recognition of G12V or, some of these ones where there have been,
Yeah.
they're rare, but there are T cells that will engage those.
Yeah. My favorite,
oncoproteins.
my favorite idea is basically, so the degrader idea is great because if you induce degradation, you have more peptides. That helps antigen presentation. And the cool thing is we haven't done yet, but you'll get this. If we had a degrader molecule, then basically this piece would look like the part that bound to RAS, but then there'd be the PROTAC version, right? So then it would bind to some E3 recruiter. So then we could make this thing not an antibody to the drug, but just to the E3 recruiter. And then you could use it for any covalent PROTAC, and you would have this strategy every time. So yeah, that's great, yeah.
Feel compelled to ask one more. But first, the CME code, oh my gosh, by memory, 44447. Okay, good. Okay, so generating anti-drug MHC antibodies was probably quite difficult. Can you make MHC-restricted display libraries that may be better for screening different drug peptide adducts?
Great idea. We haven't done that, but this came out of a naive library. So yeah, it would be hard. Those are typically very hard, but because the drug is such a big epitope, that's kind of was the secret sauce. Yeah.
All right, let's thank Kevan for a fantastic talk.